So I hope all of us are with our writing materials, our pen, and our notebook. Okay. So this is week one, more like an introduction to biology and microbiology. So we'll be kicking off with our learning objectives for this night. Now, by the end of this session, I want all of us to be able to answer questions kind of like originating from these learning objectives. So this is just what we are expected to learn from this section. Are you with me? So number one, by the end, I expect all of us to be able to clearly define what is biology and microbiology, and also to understand their distinct areas. Two, I would love us to be able to explain the origin of living things using scientific theories. I also like us to be able to trace historical origin of microorganism, master scientific method, how it's applied in biology, explore relationships between biology and fields like agriculture and medicine. Also, you guys should be able to gain an overview of biological molecules and how microbes are discovered. And lastly, to be able to identify, describe, and draw major laboratory equipment used in biology and microbiology. So we'll be going on to the next slide. Introduction to biology and microbiology. Am I audible enough? Hope you are still together.
Yes.
Okay, nice. Please, all of us will be responding, okay? So this will be more like an interactive session. This is biology. We are not just learning theories. We are learning things we can be able to apply. So first of all, when we talk of biology, I would love to share a little story. Now, when I was growing up, I used to imagine, I think I was in primary one then, I used to imagine how do my mom give birth or how my mom give birth to my last born. I'm older than my younger brother, just a year older. So I wanted a baby sister. So I'll tell my mom, I want a baby sister. And she'll tell me, no problem. She'll give birth to a girl. And every time I go to school, I wonder, how will she give birth to a girl? We have stomach just burst open one day and then my sister will now jump out. Or will probably something from the sky will just boom, deliver the little girl to her. So I was that curious. Now, this curiosity made me ask questions pertaining to life. Are you with me? So these questions are more like probing into how life began. What are the processes of life? How do organisms interact with one another? How do we give birth? How do we breathe? How should I just fall down and then blood will start bleeding? What's the process behind it? Now, this and everything put together under a systematic study process is what we call biology. Are you with me? So you can simply define biology as the scientific study of life and living organisms. So when you talk about biology, it's just you sitting down now, just like I did when I was still in primary one school, primary one, that's years back. Just like I sat down and was thinking, how will my mom give birth to this my little girl? How will my mom take in? How will she deliver? Will her stomach just tear into two? Or will she vomit my sister? Are you getting me? So I'm now making a detailed study on the process of life. Are you guys still with me? So the next thing we are going to look at
Yeah.
as well is thank you very much. Next thing we are going to look at now is microbiology. Now, let me break this word into two: micro, then biology. Okay, biology itself as well. Let's break the word into two. Let's go to the origin of biology. Biology is derived from two words. Bio, meaning the what? Bio means what? Can anyone help? Okay, I think I'll just go on. Bio means life, and then logos means study. So when we talk about biology, it's just simply the study of life. Now, let's look at this thing as well in the aspect of microbiology. Micro means something that is very, very small. You can't see with just your eyes. Are you getting me? and then biology as well. So microbiology is kind of like talking about, this is not just about organisms now, but little organisms that are what, very, very minute. Something you can't just look and see with your naked eyes. So you can define microbiology as what? A branch of biology focused on what? Microscopic organisms. That is, organisms that are too tiny that we need what? An equipment to be able to what? See them. So now, when you get all this knowledge together, as a medical student, I love all my students to ask the question why. I always encourage that. Just like I was way young and I was asking the question how, why should this be like this? How will this come to be? Likewise, I expect all of us to what, have that curiosity to know how things are. So we are now in school. We are now preparing to what, take a big step into the university. So what we'll be treating here, they're not just telling you things for the sake of you knowing them. They have what, reasons to what, doing them. Now, this course, biology, why do we study biology? Why do we study biology? Now, first of all, we study biology to us understand different life processes. Just like my analogy when I was growing up, I did not understand how my mom would give birth to my little girl. So the thing keeps bugging my mind. Now, the knowledge of biology will what, teach me how a woman what will take in. It will now teach me, okay, I think that's in your second semester, second level, second year. You now learn the process of what, embryology, how the what, baby is conceived, the fusion of male and female sex cell to give what, to give rise to a zygote, how that zygote will undergo what, different processes, and eventually to what, delivery. This is what biology is looking at. Number two as well, we are studying biology to also understand health. They used to say health is wealth, right? So we also study biology to understand how our health and well-being function. We also studied biology to understand our environment. To also have develop innovation in medicine. Few years back, some diseases were incurable, and right now there are drugs to manage those diseases or even to cure most of them. I know when I was way younger, HIV and AIDS was a very terminal and terrible disease. Once you get it, you are gone. But now HIV and AIDS is manageable. It can It can be treated to some extent. Your life can be prolonged. Now, is knowledge of biology, how all these and all these enzymes, microbes, and everything interact in your body that is able to do give us now knowledge to prolong life. Are you guys still with me? So we'll now be looking into the next slide. Origin of living things. Origin of living things. What are living things? Living things are simply anything that have life in them. At least that was what we are taught in primary school. Mr. During my own session, our teacher told us is Mr. Niger D. That's characteristics of living things. So we now want to look at how does these things come to be. My childhood was very, very fun. Are you getting me? I used to imagine a lot. I think a lot. I want to just know how everything looked like. Now, I used to imagine, how do life come to be? How do I myself come to be? The only thing I know was my mom gave birth to me. But where was I before my mom gave birth to me? How my mom even came to be at the first moment? Now, these questions, I was not the first person to ask them. There are people that was lived ages before us that asked the same questions, and they had beliefs. So one of the earliest belief, now, please, if you are writing, take notes. One of the earliest beliefs, okay, please, if you are writing, the slide is there. I can send you the slide. We'll send the slide when we are done. But there are some things I will be teaching that may not be inside the slide. So know when to take notes. And if I say you should point out to take notes on anything, please write it and underline it. It's very important. Now, one of the earliest belief about the origin of living thing is, number one, spontaneous generation. Spontaneous generation. Now, when you say something is spontaneous, it means the thing just happens. It happens randomly and almost immediately. That is, it has no precause. Nothing just led to it. it happened on its own. So one of the earliest beliefs was that life just happened. We just opened our eyes one day and then we saw ourselves. I saw my mom. I cried. And next now I was giving milk. So, but then this belief was disproved. They were like life just originated from what non-living matter, just like that. But a man named Pasteur, he disproved this what theory. As we go down, we'll see further reasons. Now, the second one is called abiogenesis. That is, life began from non-living matter under very specific environmental condition. Hmm? They were saying, now, I think most if you do read stuff, you should have heard about the Big Bang theory. Some things happened in the cosmic world, then there was a bang, and some protons and some hydrogen atoms and some things came together, and then the first molecular cell was formed and all. So they were like, something just happened. There was this specific condition on Earth, and something happened tremendously, and life was came to be. Now, secondly, there was what another theory called biogenesis. Biogenesis, that is, every form of life originates from what? Pre-existing life. Now, this is one of the theories that was proven by experiment. Every cell originates from pre-existing cell. If I should cut my hand now, the cells that will appear there, they will not just appear overnight. No, the cells that were not damaged will what develop and reproduce. They will replicate and form what more cells. At this point, I think I need to know how many of us are still following. Are you guys still with me? Please unmute your mic and respond. Are you
Yes,
still with
sir.
me? Okay, very nice, very
Yes,
nice.
Yes,
sir.
sir. Well,
All right, beautiful. Okay, so let's move on. We are 18 minutes already into the lecture. Now, after biogenesis of life, then there was these two individuals, Miller and Urey. So Miller-Urey experiment. Now, what these two did was, in their laboratory, they tried to stimulate the earliest S condition. They brought methane, they brought some gases, and then they brought it together in their lab, and they tried their best to just see if they can produce such conditions as we are early on time to know what will happen. So these people, they brought these gases, hydrogen, methane, they mixed up a lot of things, and then they initiated an electric spark. That is, they believe lightning probably struck somewhere and gave birth to and led to the rise of life. So they did this same thing in their laboratory, and funny enough, they developed what amino acid. Now, along the line, you get to understand that amino acid is one of the what monomers of protein, which is what a biomolecule. Biomolecule simply means biological molecule. There are four major, sorry, I think, yeah, four major biomolecules. Let you get there first of all. So now, the modern theory stated that life started from simple organic compounds. Now, these simple organic compounds what evolved into complex cells. Are you with me? Now, Miller and Urey experiment, Miller and Urey experiment demonstrated that organic molecules can be formed even from inorganic molecules, that is normal, methane, oxygen, and others. They demonstrated this. So this modern theory built on that their experiment to say, okay, it simply means life started from simple organic compounds. Now, these organic compounds came together and gave rise to what? Complex cells. There's also something called the RNA world hypothesis. This hypothesis says that RNA were the simplest replicating molecules. RNA, one of my molecules of life, something called the nucleotide. Okay, let me just mention my molecules of life. There is carbohydrate, there is protein, there is lipid, that's fat and oil, and then there is nucleotide. These are the four biomolecules of life. So in one of these four is the nucleotide. Nucleotide are what makes all the RNA and the DNA. So the DNA now is your whole entire genetic makeup. It carries the coding instructions for your proteins. So the RNA hypothesis was saying that RNA were the first what mutating molecules. They were the first molecule that were formed that has the ability to do what? Divide and produce many of its kind. So they now pointed out that RNAs are the what? Precursor to life. Please, if you're writing down somewhere, just underline that point. RNA are the what? Precursor to life. So let's move on. Definition and scope of biology. We have defined it earlier. We say that biology means what? Bios meaning life and logos meaning study. So when you talk about scope, what are we actually saying? Scope just means extent, areas. So when you talk about scope of biology, we are talking about what are the areas that biology cover? What are the what's the extent? What's how far can biology go? What does biology entail? So biology has covers every life form from plants to animals to humans to microorganisms. And I believe everyone here knows that humans are part of it, right? Called Homo sapiens. So biology covers all of this. It deals from what the plants that you can see, then the animals, then you yourself, and then microorganism. Microorganism, take note. Now, microorganism includes those ones that we can what, see only with the help of what microscope and the rest. Biology covers them all. So what are the subdisciplines under biology? We now have botany. Botany, which is the study of what? Plants. We have zoology, we have ecology, we have genetics. Oh my god, I really love these genetics. I wanted to actually study genetics before. Well, I changed my mind. Then there is cell biology, there is physiology, the study of function, and there is microbiology. Okay, so biology interacts with other fields, that's with agriculture, with medicine, pharmacy, biotech, and environmental science. So biology is not just in isolation. Now, when you are planting something, biology comes and tells you this is how cassava what grows. Cassava is still plants. In medicine, biology comes to tell the doctor this is how the normal body should function. That's biology and physiology. This is how it should function. When the body deviates from its normal function, we now what involve pathophysiology. So biology is no the knowledge of biology now helps us in medicine. That's why tomorrow, by God's grace, when I graduate and induct, you'll now come to my hospital and then I'll be telling you this are what happens to you, or this is what should happen and all. Are you getting me? So in the field of pharmacy as well, biotech and the rest, biology is what involved. So we can say that the scope of biology is from what? The molecular level. That's your DNA. Molecular level from your DNA to what? Ecosystem and global biodiversity. So in essence, biology starts from the very grassroots and then towards to the top. So the next slide says scientific method. And I told us that one of the things I want us to develop after this class is the ability to ask what questions. That is why. So if you have been a good student, you would have asked, how does things just come to be? How are laws formed? Okay, if I should ask you now, what's the mechanism of breathing? You say inspiration, expiration. How does it come to form? How are laws stated and all? Biolo Scientists, biologists, they just wake up and tell you this is this. They just wake up and tell you all cells originate from pre-existing cells. No, there are step-by-step processes they follow to confirm this what's law. For them to tell you that if you should cut your hand now, cell mass will not just appear, that the cells there will now start replicating to form new ones. It means that they have carefully observed it. They have questioned that their observation. They have generated some sort of hypothesis that's kind of like testing this should be what happened or this should be how it happened. And then they experimented on it. And after everything, they collected what available data and or draw a conclusion and repeat the process. Are you with me? So in all this, I can simply define scientific method as what? There are what? A systematic process scientists use to observe, ask questions, test ideas and draw conclusion. I think this is not in the slide, so you can write it down. So Scientific method are what? Systematic processes scientists use to observe. When they observe things, they ask questions. Now, they test the ideas and also what? Draw meaningful conclusion. Why? I told us scientists, they don't just wake up and make a guess and then turn the guess into law. They follow what? Step-by-step processes. So, I would now love us to be looking at this what? Step-by-step processes scientists follow. So, number one is what? Observation. Observation. And I'll be using a very simple analogy so you guys should follow very well. And we'll now be using our phone. I believe most of us are streaming this from our phone, right? So we're using our phone. First of all, observation. What is observation? Observation simply means what do you notice about a situation? What do you notice about a phenomena? Are you with me? What do you notice? Then you gather the data and then use your senses or instruments. You gather data. So first of all, imagine your phone. Suddenly you pick up your phone and you press it to power it on. Your phone did not respond. You did not see any light, no ITEL, no techno, no sound, nothing, nothing. You observed something. Are you with me? Next step is called the hypothesis. Sorry, next step is called questions. All right, questioning this. You now what? Derive question from your observation. Someone should write it down. Question. That after observation, you move to question. You now ask questions about your observation. Why is my phone not turning on? Are you with me? You have observed that your phone is not on when you press your power button. You now ask questions. Why is my phone not turning on? It will lead you to your next point, hypothesis. So since your phone did not turn on, you now start thinking, what do you think? What actually happened? Why is it that my phone is not turning on? This is what's called an educated guess that explains the observation. That's simply hypothesis. It's just kind of like you making a guess to do what? Explain what you have observed. Since my phone did not turn on, probably the battery ran out. Hmm, that's making sense. Or maybe the phone has spoiled. So are you seeing two hypotheses now? Since this phone failed to turn on, I can make, okay, let's make it three. Probably the power button is faulty. Number one is what? Probably the battery have died out, have run out. Number two, the phone have spoiled. The phone is what? Faulty. Or number three, the power button what? Has issues. Are you getting me? This is called on a what? An educated guess. Now, the fourth part, we experiment. That is, we test our hypothesis under what? A controlled condition. Since my phone failed to on, maybe the first idea was that the battery ran out, right? So what I'm going to do is I'm going to quickly bring my charger, plug it in the light, and what? See if it will start charging. Now, okay. Now, once I plug it in to see if it starts charging, I will now move to the next step, data collection. That is, record your what? Observations, quantitative and qualitative observation. You now write down what did you observe. When I plugged it in, what happened? Did it show light? Did it not show light? Was it just like that? What happened during the process? Are you with me? Then, after this data collection, you move on to the next phase, analysis. Now, it's in this analysis that you now start thinking. You now start putting in your data together. Now, when I plugged my phone in, what happened? Did it start charging again? Did it show light? Did it respond? Did it not respond? Now, after it, you draw your let's continue being what and positive. Let's see now when I plugged it in, my phone showed light charging. Are you with me? I'll now draw my conclusion from my what observation. After charging it and I powered it on, my phone was showed ITEL A70 and powered on. Are you with me? I'll now draw my meaningful conclusion. Okay, when the phone was not powered initially, so I now charged it and finally it is now on. So what is the results? Which hypothesis was right? Remember, we developed three hypotheses, right? Number one was that probably the phone is dead. The second hypothesis was that the phone is faulty. And the third hypothesis was that the power button the power button is what? Faulty or malfunctioning. So I'll now come and draw my what conclusion. Since I charge this phone and I can power it on now, it simply means that the problem was what? From the battery. Are you with me? And now we'll now repeat it. You can now bring other phones. Now maybe bring like 50 more phones that are dead, charge it to make sure that it's not just coincidence or just probably random something. And bring 50 more phones that are dead, charge the battery, power it on, do it more and more and more. That way you now build from what you now build it into what a law. That's why you can now come and tell someone, probably your grandma in the village. Your grandma now probably she use her phone for the next 3 days and see you after probably next 2 weeks. She'll be like, my phone has spoiled. This Umaza, they have spoiled my phone for me. You now look at your grandma and tell her, your phone did not actually spoil. What happened is that you ran out of battery. Why are you making this assertion? Is because you have tested it repeatedly to do what? Prove this point. Are you with me? I believe we are still together. Now, okay, let's just say that during your conclusion, you charge the phone, you on it, it still did not respond. What are you going to do? You now what? Reject that hypothesis, number one hypothesis, meaning the fault is not from the battery. We'll now go back to number three. Are you still with me? You now go back to number three and ask that question again. Why is my phone not on? Number one, our first hypothesis, probably battery is faulty. We have rejected this, right? So the second hypothesis is that maybe the phone is what? Faulty. That is, the phone has spoiled. And let's now say, okay, we took the phone, we charged it again. It responded. It showed light it is charging. After everything, we tried owning it. It was not yet owning. This shows that the phone have not totally spoiled. Are you with me? It means that the phone is still good to some extent. So coming down again to conclusion, we'll reject this hypothesis. We'll now return back again to question 3, to number 3, and ask again, why is my phone not on? Are you with me? Now, when you ask the question again, our third hypothesis was possibly the power button is faulty. And now we'll take our phone to the phone repair shop. And when we reach there, we'll now give it to them. They will change the power button and all. After it, you'll be handed over your phone. When you press it and then it turns on. Are you getting me? You now come and draw your conclusion and say, okay, the matter was that the power button was faulty. This is just the simple steps that scientists follow every single time to bring to conclusion what they have in mind. Are you with me? Okay. We are 34 minutes into the lecture, so let's just run down again. Hypothesis, data collection, and application. Okay, this is just an example. Sunlight affects plant growth. We'll now say, okay, observation. When you put a plant in the light, now you are watching it. Okay, imagine you saw a plant outside doing well, growing very tall, and then the one inside is somehow bending. You ask yourself, why is this thing bending? That's questioning. After it, you now draw a hypothesis. Maybe it's bending towards a light source. Hmm? So you now conduct an experiment. You will now make another plant, keep it near your window, close the whole curtain and all. Just probably cut a slight opening for sunlight to be entering in. Now, after some days, you return and collect your data. You now look at the thing and say, okay, how is this plant doing? If you notice that the plant is bending towards the sunlight, you now come and draw your conclusion and state, okay, since the plant bends towards the sunlight, it means that this plant is what? Growing towards what? Sunlight. That sunlight affects the direction of what? Plant growth. I hope you are with me. Now, you now repeat this same hypothesis, this same experiment countless times under what? You can now repeat it countless times. Now, when is what proven beyond doubt, it becomes a law that what plants tend to grow what in the direction of sunlight. So, okay, you mentioned data earlier. There are two types of data. Number one, qualitative data. When you talk about qualitative, I'm talking about descriptive things you can see, things you can qualify with adjectives, something like color, shape, behavior. Now, okay, imagine that I do not pour my plant water for 3 days, what will happen? Probably after 3 days, the plant will shrink and everywhere kind of like die. Hey, now that is what descriptive. The second type of data is what quantitative. It's just you taking what numerical number, length, weight, temperature. Okay, you now come now measure the length of the plant after 3 days, or you check the weight, or you measure the temperature of your room. These are what's called qualitative data. Now, what are the tools used for what? Experiment. Look at the very common tools you can easily see in your laboratory. Number one is your microscope. You can't study anything microbiology without your microscope. Number two is ruler for taking measurements. Number three is the scale. You use your scale for taking the weight of something. Number four is the what thermometer, and number five is the what stopwatch. These are very simple things you should see in your laboratory. Even you as a student, you can take them in. When you observe natural phenomena, you can what start up your process, scientific process immediately. Okay, so what's the application of these steps in real life? Number one, agriculture. Okay, I don't know how many of us have seen where parents plant something and use fertilizer. But I can remember a scenario during my secondary school. We are told my elder sister was told to plant something and use fertilizer on it. Funny enough, we used fertilizer on the plants, but we put the fertilizer very, very close to the root of the plant. After 2 days, the plant dried up. Are you getting me? The fertilizer killed the plant instead of helping it. So in this scenario, we can now draw up a hypothesis. You can now draw up employ our scientific method to study why did this plant die. Fertilizers are meant to boost plant growth. Why did it kill it? Are you getting me? Now, when you put it to test, you are what applying this knowledge to agriculture as well. When you have gotten okay, I should not put it so close to the plant's root at all. You can now start testing out which fertilizer is actually the best for cassava. You try this one, try the other one. I'm not a farmer, I don't know the names of fertilizer. My specialization is just school and all. Are you getting me? Now, if you have a father that is a farmer, good and fine. Or if you know someone that farms, you can ask them which fertilizer is best for cassava. They will tell you. Which fertilizer is best for legumes? They will tell you. Now, the next field, that's my field actually. So, application of biology in medicine. You use it in testing of new drugs. Hmm? Okay, during the COVID-19, there was what mass production of some drugs to what help cure COVID. Now, biology tells us what use this process, they will come and bring in patients, observe the patients. After some time, they will come, they will inject this drug. Now, they will what create up observation, questions, hypothesis, experiment on it, then collect data, and then draw conclusion. Did this medicine work out? Did it not work out? Why did it work? Why did it not work? Are you guys still with me? Then environment and the rest. Okay, biology and other field. I want to tell us that biology actually is not working in isolation. It does not just exist on its own. No. Biology is also what integrated into various fields. I've mentioned how you can employ it in agriculture, right? You can use the knowledge of plants and pest control, even soil health to do what? Improve your what? Agriculture, improve the what? Plants. See, the world population keeps growing, but the land available to plant is fixed. Are you getting me? The knowledge of biology will now tell us, see, between this species of beings and this species of beings, this one will do very well. This one is easily attacked by this pest. This one is not. Now, how can I be able to control my plant so pests will not come and infest on it? Now, the knowledge of biology will help us to produce food that will very, very enough to feed us human life. Take on the aspect of medicine, thirdly, in pharmaceuticals, environmental science, biotechnology, that's genetic engineering, cloning, and biofuel production. So moving into the next slide, history of organic molecules. Okay, please, so far, is there any question? Any question so far? Are we still together?
Yes.
Okay, Alexander. Who else? Who else is still following the class?
We're still together. So.
Okay, Pablos, I see you. Flourish, I see you here. Chinima, Christian. Boma. Okay, so no question so far, right? All right, let's proceed. History of organic molecules. I told us earlier that in one of the hypotheses, sorry, one of the origin of life, it stated that life what originates, one of the origins of life, that life what originates from what biomolecules, right? Okay, so organic molecules. So now we're looking at this what organic molecules. Organic molecules are what carbon-based compounds essential for what life. We cannot go into chemistry now. In university it's called ICH, industrial chemistry. You can't be going into it now. I believe we thought about it in separate course. Now, there are what? Four major types of organic molecules for life. They're also called biomolecules. Please take note. Anywhere you hear about biomolecules, we are talking about these four major molecules essential for life. Number one is what? Carbohydrates. Number two is what? Proteins. Number three is what? Lipids. Number four is nucleic acid. Hmm? Now, these four are what? Major biomolecules of life. Now, what are the history of these biomolecules? From the Miller and Urey experiment, remember I told us that these people, the way they experimented is they created an environmental condition close to the one we had earliest form of Earth, and applying lightning and some techniques, they were able to what? Produce amino acid. Amino acid is a what? Precursor, is a monomer of protein. Monomer simply means what? A single basic unit. Protein is a polymer, that is made up of what? Repeating units of what? Amino acid. Are you getting me? Okay, when we talk about poly means many. Poly means many. That ma, M-E-R, ma simply is a Greek word meaning parts. So when you talk about polymers, it simply means many parts. Mono means single, right? Ma means part again. When you talk about monomer, it simply means single part. So likewise, when you see proteins are what? Polymers of amino acid, it simply means that polymers are what? Made up of many parts of amino acid. So amino acid comes together to what? Form proteins. Hope you guys are still together. So let's come back to biology. So these experiments suggest that organic molecules can form under natural conditions. Remember, they say that life just existed, that's spontaneous word theory, and life just boom, we saw ourselves and we began life. No. These two, now their experiment proves to us that there are specific conditions on Earth that least gave rise to these words, biomolecules of life. Now, if these biomolecules of life should come together as well under specific conditions, they can give rise to a cell. Hmm? So the experiments form the basics of what organic molecules, that is, organic molecules giving rise to life. So their importance, it formed the basis of the first cell and life as we know it. So the history of microorganisms proper. History of microorganisms. So we'll be looking at how these microorganisms, how did you get to know about them? Hmm? Because first of all, they say seeing is believing. But how then do some people now leave things that are visible and start probing into things they cannot see? How can you tell me now that something as small as bacteria can put me in such a condition that I cannot stand up on my bed? Or how can you tell me that something I can't even see, and then they call it COVID-19, that I'll be walking along the air, probably if the air is poisoned with the pathogen, if I breathe it in, I'll start suffering from that virus. So now we want to look at how we came to establish that there's something like microorganism. So this dates back to 1665. Robert Hooke. He was my great grandfather. I was there with him in the spirit when he was observing these cells in a cork. So I don't know how he got his hand on a microscope then, but with the help of it, he observed cork cells. but these cells were dead cork cells. So when you observe, you notice that there were small compartments in it. So he was like, oh, this resembles small rooms, compartments. He called them cells. But since these plants were dead, so that was all he saw. So now, around 1675, he's my uncle from my mother's side. You know, my mom. How were we even related again? Okay, but I just know he's my uncle from my mother's side, Antonio Van. Look, are you getting me? So he's from that my mom's side. As well, he was observing some things again, and he discovered microbes using a very simple microscope. He observed that there is something called bacterial and protozoas, and he named them. Please find somewhere and write it down. Animalcules. That's ANIMALCULES. So Antonian Anthony is what he is called the father of microbiology. Write this down as well. Anthony is called the what? Father of microbiology. He is the first person to observe living microorganisms. So we write now three things from this young man. He is what? The father of microbiology. He is also the what? First person to observe living microorganism. Are you with me? So let's move down to Louis Pasteur. Louis Pasteur, he was my very good friend then in high school. Just that I don't really know his problem. He likes milk. The young man, he loves milk and wine so much. So I remember one time after secondary school, we are going home. Then his father drinks a lot too. But then his father noticed something. His wine spoiled. So the father started blaming him and was like, not him actually. The father started blaming the village people and they poisoned his wife. Something happened, witchcraft, and the wine spoiled. And the father lies to drink. So this my friend then, Pastor, he picked interest. He wanted to know why the wine spoil. And his father likes wine. He himself, he likes milk. I don't know, something about liquids runs in their bloodline. So he himself, he likes milk. And when he started studying about milk, he noticed something as well. Now, he brought some milk, he boiled it, I think, under 72 degrees Celsius for like 15 seconds. And then he covered it airtight. So after weeks, the milk remained the same. The milk did not spoil. So he was confused. He was shocked. He developed a hypothesis. Possibly, there is something inside the milk that is causing this milk to spoil. So he now heat up the milk again. Remember now he sealed the tube, making sure that there was no way any other foreign no air, no other foreign body can enter inside. Are you with me? Now he kept the milk. And funny enough, the milk lasted for weeks without spoiling. And he drank the milk happily. So probably he did it again, left it open. Now, when something is open, is in communication with the environment. Microorganisms, as you know now, can now enter and live. But then they did not know anything called microorganism. So after some days, the milk spoiled. So he established what is called the basis of germ theory. Please write this down as well. So Louis Pasteur disproved the spontaneous generation. That is, he disproved that things just happen, that microorganisms just exist. No, he cancelled out that thought. Remember, I told us that in one of his experiments, he covered the he did it in a closed environment. That is, the tube containing milk was covered. That way, there was no introduction of any other thing from the environment, and the milk remained like that. So, and the microorganism did not just generate within the milk. Now, in another experiment, he left it open, so the milk can now interact with the environment. And in so doing, he discovered that the milk spoiled. So he disproved what is called the spontaneous generation. That is, microorganisms don't just come out and be. And also, he proved that microbes is the cause of fermentation. Why the milk spoiled was because the milk fermented, and the milk not just ferment because that's the nature. No, it's fermented because there were microorganisms inside it. So now I want you to write down two things concerning Louis. Number one, Louis Pasteur disproved the spontaneous generation. Louis Pasteur disproved the theory of what spontaneous generation. Now, the second point is that Louis Pasteur also laid the basis of the germ theory. Now, you can put brackets. Okay, let me come again. Louis Pasteur disproved the spontaneous generation. That's the theory of spontaneous generation. The second point, full stop. Now, write again. He also formed the basis of what? The germ theory. Now, in bracket, germ theory, that is, diseases are caused by specific microorganism. That's your bracket close. So let me come again. Bracket open. Diseases are caused by specific microorganism. Bracket close. So what this germ theory simply means is that the microorganism responsible for causing HIV is different from the one responsible for causing tuberculosis. Microorganism responsible for causing syphilis is different from the one responsible for causing gonorrhea. So if someone is infected with gonorrhea, it does not mean that that microorganism will now cause the person to have syphilis, HIV and AIDS, COVID-19 and the rest. No, that's the basis of germ theory. So let's go down. Robert Koch. Ah, damn, this my guy. He one of my friends in secondary school there. Just that I don't know how they are all older than me. So he now built on what Louis Pasteur said. You know, all of us went to the same secondary school together. So when this guy came in the following day to school and was sharing, he was shouting. I was like, guy, have fun now, calm down, calm down. He was like, I don't discover new thing. Oh. And after everything, Robert Koch went home and started experimenting. So he discovered something. There is a disease called anthrax. That a specific microorganism called Bacillus anthracis was responsible for causing anthrax. So he built on that germ theory to prove it that specific microbes cause specific disease. All right. So what I want to take away from this is that Robert Koch established the germ theory. In brackets, specific microbes cause specific disease. Bracket close. I'll repeat. Robert Koch established the germ theory. In brackets, specific microbes cause specific disease. Full stop. So moving to the next point, Alexander Fleming. He was one of my best friends. Young man is very, very bright. He discovered penicillin. If I've heard about antibiotics, you know that he's actually life-saving. So he revolutionized infectious disease treatment by discovering penicillin. Okay, so let's run. Modern microbiology studies structure, function, genetics, and the use of microbes. So this will be one of the last thing for this class. Then we'll ask questions. Since we started, okay, we ran through a lot of things, and we are now nearing the end of the slide. This is now the practical section, biological/microbial lab equipment and drawing. See, we scientists don't just use our head for everything. Even you yourself, I believe most of us are writing, right? Exactly. So there are some equipment, some materials, some instruments you also use to be able to do what? Help us collect data, to help us learn, to help us pick samples, to help us carry out our virus experiments. So I'll just take us through the essential ones, and then I'll give us an assignment. When we come for the next class, I'll ask how many of us did it. So one of the essential lab tools we cannot do without in our laboratory, especially when it comes to microbiology, is the microscope. The microscope. The microscope. Hope you are seeing the diagram here. Microscope. So if we are going to study anything that has to do with everything that has to do with very small organisms, we must use a microscope. So see that the microscope is an instrument used to do magnify microorganisms. There are microscope, there are instruments that use they help us to enlarge our scope, our vision scope. our human eye, oh my God, used to fail us a lot. There are so many things you cannot even see spiritual things. Talk more of things that are too later. Are you getting me? So the next instrument is the petri dish. So the petri dish, that's for, I don't know how many of us are aspirants for lab science. That's med lab scientists, med lab science. In med lab science, they know the petri dish is used for culturing microorganisms. That's culturing simply means what? Culturing simply means creating what? A growth environment for microorganisms. So, number three, test tubes. One of the things I remember from secondary school, out of all the practical classes we went to, was if you break one, you buy five. So whenever you hear about test tube and pipette, it's just if you break one, you buy five. That's our chemistry teacher for you. So the test tube is used for what? Holding samples and reactions. So next is the Bunsen burner. I think I have a picture of it as well. The Bunsen burner. I believe most of us have seen this equipment here before. It's just a very simple something. So the Bunsen burner is used to what? Provide flames for sterilization or for heating. Now, the next one on the slide is what? The incubator. Incubator. Now, whenever I heard the word incubator, what comes to my mind is eggs, right? Chicken eggs, you incubate them until they hatch and the rest. Also, I want to make a notice as well that children that are born before time, they are also placed in special incubator to provide that temperature needed for them to thrive. Hmm? So that being said, you now define the incubator as a what? An instrument that provides optimal temperature for microbial growth. Now, lastly, the pipette. So the pipette is used to do what? Transfer liquid accurately from probably your beaker to your conical flask or wherever else. So how do you do it? Normally you place it, then you suck it into the what? The mark and then use it. They have 25 ml and they have 20 as well. So okay, I've reached the end of the class. This is just a drawing tips. If you want to draw something, use a very sharp pencil. I believe you must have heard this countless times. Use a very sharp pencil, label all parts clearly, add scale and title, use clear lines, and of course, you don't shade in biology. Okay, so let me just conclude. So this week we have been able to lay the foundation of biology and microbiology. That's what we did today. We laid the foundation of biology. I told us what biology is all about, the study of life, and microbiology is all about studying life at microscopic level. Also, I showed you how you can employ a little systematic step-by-step process that study into what life is of how scientists does it. It's called scientific method, right? I use my phone as an example, and we also use a common plant as an example, right? Okay. So next week we'll be looking at we'll delve into cells, building blocks of life, and also to prepare to explore their structure, function, and how they sustain life. Okay. So one more thing. What aspect of biology surprised or probably fascinated you the most? What course you want to pursue in this medical aspect? Or I don't know why I feel like most of you should just join me in the medical field, but I know any one of us here must have something about the medical aspect. So this is the end of the slide. I'll now want to take questions from us, and I'll also ask my